everyone, and welcome to episode 113 of Cell and Gene, the podcast. I'm Erin Harris, and today I'm excited to welcome Dr. Thorsten Gorba, VP Process Development at Aspen Neuroscience, to the pod. Dr. Gorba and I dive into a really fascinating topic, how Aspen is raising the bar in cell therapy manufacturing with the integration of machine vision, AI, and machine learning. Dr. Gorba and I talk about how these technologies are transforming the way scientists evaluate iPSC colony quality, which will help move past the limitations of manual cell assessment and toward more reliable, scalable manufacturing. We talk through how all of this impacts consistency in starting materials. We unpack the regulatory angle, and we look at the broader potential for advanced analytics across the cell and gene therapy space. So thanks for tuning in. And here is my conversation with Aspen Neuroscience's Dr. Thorsten Gorba. Enjoy. Thorsten, thanks so much for being here on Cell and Gene, the podcast. I'm happy to have you.
Thank you, Erin. Thanks for inviting me. And yeah, great pleasure to be here. Yeah. Mm-hmm.
Good. Well, thank you for your time. You and I are going to get into how Aspen leverages AI and machine learning, or ML, to accelerate the development and improve precision and all that the really important things in order to tackle the challenges in personalized cell therapies for Parkinson's et al. And so, but before we dive into all that and really get into the nitty gritty, tell us a little bit about Aspen Neuroscience.
Yeah, certainly, Erin. So at Aspen, we are developing autologous induced pluripotent stem cell-derived medicines for diseases with high unmet need. And our lead program is in a phase 1/2A trial for moderate to advanced Parkinson's disease. And really importantly, so the autologous approach, which is really something that Aspen is specializing in, eliminates the need for immunosuppression, which can be really problematic in an elderly patient population like Parkinson's patients. And so for this particular treatment, we produce autologous dopaminergic precursor cells, which are transplanted directly into the putamen of the patient's brain. The goal is for these cells to engraft and relieve the motor symptoms of Parkinson's disease. We have already demonstrated the safety of this therapy and have presented data at renowned international conferences, for example, ISSCR. So what differentiates us from other developers, as you said in your introduction, is that we are very heavily leveraging genomics, bioinformatics, artificial intelligence, and machine learning to provide the highest quality cell therapy products for every patient, which is specifically important when you do an autologous therapy where you derive cells personalized from each patient. And yeah, further, we are also automating our manufacturing processes to increase the production capacity and reduce the costs of the manufacturing process to reach more patients who could benefit from our cell therapies.
Good, thanks. Okay, well, thank you very much for that detailed background of Aspen Neuroscience. And like I said at the top, that we're going to focus pretty heavily on AI and machine learning throughout our conversation. And so I would like to talk a little bit about how Aspen Neuroscience is in fact leveraging machine vision and AI or ML to assess the iPSC colony quality. So talk us through a little bit about what that actually looks like and how these technologies are being applied and why they're so important for, like you said, advancing the manufacturing aspect of this.
Yes, certainly. So at Aspen, we are combining machine vision with AI ML with robotic cell processing and automated liquid handling. And for that, we use the Celligent cell culture platform. That is a platform that is not widespread in the industry. It's something that we have co-developed. So the Celligent platform is really co-developed by Aspen and implemented in partnership with a company called CellX Technologies, who are a young startup company. And we have really grow to great lengths to support them and bring them along and help us to develop the platform that's for our specific needs for an autologous process. And they are a spin out from the Cleveland Clinic. So the first really important use application is what pluripotent stem cell culture experts called weeding. This means that after the reprogramming, emerging iPSC colonies, they need space to grow to a large enough size and for their quality to be assessed so that they can then survive being mechanically picked and expanded after passage. Hence, for your budding iPSC colonies to grow and thrive, the fibroblasts surrounding them must be removed. And so traditionally, this is done by skilled operators who sit on the laminar flow hood and have an open culture plate with uh, emerging iPSC colonies under a microscope, and then they use a pipette tip or similar to scratch away the cells that are in the way for the colonies to grow and expand. So yeah, unfortunately, this kind of treatment so amounts to a culture shock with the cells sitting outside the incubator for a really long time. And in addition, there's also significant risk of contamination due to the extensive manipulation in the open plate. So we have fully automated this process while keeping the cultures in the controlled environment of the Celligent platform all the time. So, and after automated imaging of the cultures, our AI ML software identifies the emerging iPSC colonies, and it does so outperforming the human eye of the skilled operators. It can see colonies that a human operator would miss and defines the weeding paths around them. So then the weeding is automatically executed by the Celligent's robotic instrument, and when the colonies reach sufficient size, the Aspen Vision software will then assess their quality by the morphology of the cultures and rank order them for automated, gentle, shear stress induced, but very effective colony pick passaging to establish high quality clonal iPSC lines.
Interesting, fascinating, actually. Um, now, okay, two things. Traditionally, so we know that iPSC colony evaluation has been both manual and subjective, but as you've just explained, this is becoming very thorough and automated. How does machine vision improve both subjectivity and that manual aspect of it? And then perhaps if we can just give a very quick definition of machine vision.
Yeah, so machine vision really is that it's like the self-driving car. So you have a computer system that has is aided by a camera and is then through enabled by AI or machine learning able to understand what it sees and act accordingly. So that would be the definition of machine vision. Machine vision really only has benefit if it's combined with AI and machine learning. So what we do, the machine vision and the AI/ML software analysis guides us and informs the automated process execution. And this takes both the subjectivity in cell culture quality assessment and different manual skills levels of human operators out of the equation. So this results in a consistent and reproducible process and improves scalability in two distinct and very impactful ways. So firstly, the robotic culture automation that runs 24/7 can achieve a much higher patient lot throughput with a smaller clean room footprint than manual culture. Yeah. And secondly, something that is most people really don't think about is the use of machine learning reduces also the need for skilled operators who need to be trained up to a year before they reach a fully sufficient level, both from their judgment and from the manual skills. And this mitigates the often overlooked bottleneck of scaling human stuff and keeping up with operator training and qualification, especially if you really move from thousands maybe to tens of thousands of patients. So it will be impossible really to find enough stuff and keep them all trained. So we really have to automate not only the execution, but also the 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 judgment and assessment of the cultures.
We talk about variability, batch to batch variability, starting material variability, um, frequently on cell and gene. And so I would love to hear from your perspective, you know, it how is it a persistent challenge for your team, and how have you used AI and ML to improve upon that from a quality assessment perspective?
Yeah, variability of autologous material is always a challenge, but autologous isn't equal autologous, so there are subtle differences. So while the variability of starting material is also a consideration for autologous iPSC-derived cell therapies, it is somewhat buffered compared to autologous T-cell therapies that mostly come to mind when people talk about variability of starting material. Because in case of T-cell therapies, the patient age and the treatment history can really greatly affect their efficacy. So the reason for that, while it's different if you have an autologous iPSC-derived product, is that pluripotency reprogramming acts as a great equalizer that really erases epigenetic march of aging and pathology. That means that even so our Parkinson's patients population is aged, the autologous neurons they receive, which are iPSC, are indeed young and healthy. And as I said, with all the tools we're using further, we go to great lengths to ensure that we derive very high quality iPSC from every patient. And so, for example, in addition to the standard flow cytometry pluripotency marker presence that is usually done, our iPSC lab must also pass an RNA-seq data trained machine learning algorithm assay that's called pluritest to confirm their pristine pluripotency and differentiation potential. So pluritest was originally developed in the laboratory of Aspen co-founder Dr. Jean Loring and has become a widely adopted quality standard in the research community. So that shows that we are not only using the machine learning really for process control, but also for using very sophisticated genomics-based release assays. Yeah. And so, in addition, so we have demonstrated throughout our process development with the Celligent platform that our machine learning algorithms consistently produce iPSCs meeting our high quality standards. If we are producing high quality iPSCs, this leads to high quality dopamine neurons as well. But to completely make sure of this in the final drug product QC step, we introduce another RNA-seq-based machine learning algorithm that we call NeuriTest, which is done to confirm similarity of the released lot to dopamine neurons precursors that were shown to be efficacious in the past.
Good. Okay. Thank you. I'm curious to know, in addition to all of this, how you've worked with the FDA and from a regulatory perspective, excuse me, how have you been working with the FDA from an evaluation standpoint to encourage the use of AI and ML tools? What have you run into? How have you worked together? What kind of best practices have you
Mhm.
worked together with the FDA on so far from both manufacturing and quality control perspectives?
Yeah, so that is an evolving field, and really the hard truth is, as a developer, so regarding regulatory buy-in for use of AI and ML in manufacturing quality control, you really have to forge your own path. So agency like the FDA will give you a rough idea of what they expect, but you will have to define the strategy of how to get there by yourself, yeah, and then obtain their approval for what you are proposing. So it always has to be an iterative process. Yes, you need to read the guidelines and try to make the best sense of it, make a proposal, and then run it by the FDA and see if they agree. But in general, so your path can be easier if you're able to use explainable AI, which is also called XAI, such as machine learning algorithms, which are fully transparent and have no black box component. So therefore, but unfortunately, yeah, therefore, if your task is too complex and you do require deep learning, making it a non-transparent model, then the burden of validation will be greater. However, use of black box models can be acceptable in cases in which transparent and interpretable models show unsatisfactory performance. And in a very general sense, as with many aspects of CMC, the bar will be raised with the level of assessed risks. For example, risk to the successful outcome of a manufacturing run, which is mostly a business risk, will have lower hurdles than risks to product quality that could affect safety and efficacy outcome for the patient.
Okay, thank you. I want to talk a little bit about kind of a forward-looking, forward-looking statement. Where we kind of see AI ML moving with regard to not only iPSC colony selection, but even beyond that. So, what do you think, and where do you see AI and ML and machine vision making a
Mhm.
near-term impact?
Yeah, well, one very obvious use case that really applies to all adherently proliferating cells, not only iPSCs, is automated machine vision assisted confluency assessment of the cultures to guide a feed or passage decision and then initiate passaging in an autonomous automated fashion if needed so that the cells can be passaged really when the cultures reach the right stage and is not dependent on when operators are present, when operators are able to look at the cultures. It can really also then increase the quality and uh, and the yield of your cultures. So, and at Aspen, we are planning to use this application for our fibroblast starting material expansion on the Celligent automation platform as well.
Great. Okay. Finally, before we wrap up, would love to hear about what you think about the future where AI systems not only monitor, but also help optimize and even self-correct cell and gene therapy manufacturing processes in real time.
I think it's really not that far away. The future is now, and I think the first optimizations applications coming back to the risk and the hurdles will be in process development, where as much as possible in process monitoring and cell quality outcome data will be collected to then generate a model of the process. And this model can then be used to predict the potential impact of manufacturing changes in silico. And when you have this prediction, then you can reduce the number of really costly wet lab experiments than you have to do in your process development in NSEP's group. And that will help both bringing the workload, the burden on your teams, and of course the costs. Often you have, if you do stem cell cultures, you have very costly reagents. So doing a lot of that work in silico and then only execute the really the top experiments that you need to confirm what your predictions are will be really helpful. And yeah, and then as stated earlier, if it's not process development, so for application that manufacturing process decision in an autonomous fashion or drive quality control, such as real-time release, the risk will be seen as higher, and the expected proof of adequate and reliable model function will be much more stringent.
Sure, sure. Makes perfect sense. All right. Well, that brings us to the end of our episode featuring Aspen Neuroscience's Thorsten Gorba. Thank you so much, Thorsten, for your time. This was very insightful and just a wealth of information about the impact of AI and ML on the cell therapy progress for Parkinson's.
Yeah, thank you very much, Erin, for inviting me. It's great fun to pioneering this new technologies really at the edge of science and sharing our experiences with the field in general. Yeah.
And thank you, listeners, for tuning in. Be sure to visit us at cellandgene.com and to subscribe to the podcast so you never miss an episode just like this one. And we'll talk to you soon.